Welcome to Chapter 14, Part 1, Principles of Disease and Epidemiology. This chapter fulfills objective number 4, to provide students with knowledge to gain a basic understanding of the interactions and impact of microorganisms on humans and the environment. Topics will include interactions between microbes and their host, microbes and human disease, environmental and applied microbiology. Specifically, we're going to be hitting interactions between microbes and their host, microbes and human disease. In Part 1, we're going to talk about pathology, infection, and disease, normal microbiota, etiology of infectious diseases, classifying infectious diseases, and patterns of disease. In Part 2, we're going to talk about spread of infection, nosocomial infections, emerging infectious diseases, and epidemiology. Let's discuss a few terms that are going to be vital for you to understand the material in this chapter. First off, this chapter deals an awful lot with pathology, which is the study of disease. It includes 3 subsections. Etiology, which is the study of the cause of the disease. In microbiology, we generally go with those diseases that have an infectious etiology. Those diseases that are caused by microorganisms such as bacteria and viruses. Now, we also sometimes include cancer, which sometimes is not caused by an infectious etiology. It's caused by mutations in your cells, but we're finding that an awful lot of cancers are caused by viruses as well as these mutations. Also included in pathology is pathogenesis, the manner in which a disease develops. We're going to be talking about some of those things in a general fashion. And then thirdly, pathology is the study of the structural and functional changes that occur in the host as the disease progresses. Now lastly, let's talk about the difference between infection and disease. Infection is invasion or colonization of the human body or parts of the body by microorganisms. Now, disease is structural and functional changes that occur from infection. So you can have infection without disease. Technically, you are infected with all sorts of microbes all over your skin and in your gut, but it's not, generally speaking, causing disease. In fact, you want them there. So, remember the difference between infection and disease. In this next section, we're going to be discussing your normal microbiota. We're going to talk about how you get it, the relationships of your microbiota to you and to other microbes. We're going to talk about opportunistic microorganisms and cooperation among microorganisms. Now let's talk about how you get your normal microbiota. If things work how they should, before you are born, you do not have any bacteria, yeast, etc. on or in you. You're composed of just your cells. Now, in the process of normal birth through the vagina, you start picking up skin bacteria from your mother. Then, as you're handled by the birthing staff and your parents and various relatives, you start picking up more and more microbiota. You pick them up from your family, generally speaking. Now, let's talk about areas that have normal microbiota. Anything that has contact with the outside world is going to have a normal microbiota. So your skin and your gastrointestinal tract have constant contact with the outside world. So you have an awful lot of bacteria on and in you. Okay? So your skin's covered with microbiota, your gut's covered with microbiota. Now let's talk about your respiratory tract. As you breathe in air, microbes are going to come in. But you're set up that you try to keep the areas of your lower respiratory tract microbe-free. In fact, this is a good time to bring up transient microbiota. Transient microbiota are those microbes that are there for 6 months or less. If they're considered resident microbiota, they're there for 6 months or more. They've set up the lease and they're renting the property. All right, so here, my last picture that I brought up, you see some ciliated cells. So because your respiratory system is kind of a dead end, it's a cul-de-sac. You breathe in air and you breathe it back out. You really don't want microbes setting up house at the end of that cul-de-sac. So these ciliated cells, when you breathe in dust, microbes, whatever, move everything up and out. Okay, so you shouldn't have any microbes living for long periods of time in your lower respiratory tract. Your upper respiratory tract is a whole different situation, but microbes are constantly moving in and out and being pushed out of your upper respiratory tract. Other areas that are a blind dead end, okay, your urinary and genital tracts. The urinary system goes from the kidneys down through to the bladder and out through the urethra. Well, microbes colonize the outside opening of the urethra and oftentimes a short distance up, but because the flow of urine is one way, we generally keep that area flushed out. Now, if you do have microbes that move up into the bladder, you've got a bladder infection, not a good thing. If they move up to your kidneys, you're really in trouble. Now on to the reproductive tracts. Those are also blind ends. Things are moving one way. They're moving out. And you also have ciliated cells that move mucus that traps microbes. Okay. Now, the benefits of microbiota. They've done studies with animals that have been born cesarean in a sterile environment, so we're able to keep them sterile. And we generally have a control group that spend their whole lives sterile with just their own cells. And then we have other lab animals that we introduce one species of normal microbiota at a time so we can see what the changes are. What they've found with lab animals that they've kept abiotic, that they don't have a normal microbiota, they have a harder time getting the calories from their food as opposed to those organisms that have gut microbiota. They also have a higher requirement for vitamins. Turns out, at least in humans, E. coli provides most of our vitamin K. So you're getting some of your vitamins from your little friends in your gut. Also stimulates the immune system. Lab animals that have been kept from developing a normal microbiota have a very weak immune system. It makes sense. If your immune system isn't exposed to microbes, they don't develop the antibodies and they're not ready to go. So those are the benefits of your microbiota. Now, we've just finished the human gut microbiome project, and we're working on seeing what these species are and how they benefit us. There's indications that depending upon what your normal microbiota is, it can either protect or lead to various diseases. So we're at the beginning of some wonderful and interesting information. Now on to some terms that we use when we describe our relationship to normal microbiota. All relationships where you have one organism living in close association with another is called symbiosis. Okay. Usually when we think of symbiosis, we think of the first two where there's benefits, but symbiosis includes parasitism. You're familiar with parasitism. Parasitism is where one of the organisms, the host, is harmed, but the other organism, okay, usually the microbe, is benefited by this relationship. You're familiar with that one, but it's still symbiosis. You've still got two organisms that are living in close association. Now with the other two, this is where, well, let's go with commensalism. Commensalism is where you have two organisms living in close association. One is benefited, but the other one is not harmed. Okay. Now with commensalism, most of them get moved into the category of mutualism, where both partners benefit. Okay. With some microbes in our microbiota, there's an obvious benefit to the host, the human. Now, the benefit to the microbe is also obvious. We keep them warm, we keep them fed, and keep them nice and moist. I'm talking about gut bacteria, of course. This would also apply to upper respiratory bacteria. That would be mutualism. Now with commensalism, it's kind of like we can't see an obvious benefit to the host. Seems like these microbes are freeloaders and they're not hurting anything. But oftentimes, these commensal organisms, once we figure out what their subtle benefits are, get moved into the mutualistic category. Examples of this would be the staphylococcus that are growing all over your skin. No obvious benefit. But they've found that if you don't have high enough numbers of these staph species of bacteria, then that leaves you open to various skin diseases. They're taking up space so that pathogens can't attach to you. So that would be an example of a commensalistic organism that gets moved into the mutualistic category. Now, because of this relationship between our microbiota and us, as we've developed an understanding of it, we've developed probiotics. This is the idea that especially after you've had a series of antibiotics that's killed most of your normal microbiota, we want to replace them with commensal and mutualistic symbiotes, i.e. friendly bacteria. So you may have heard of this, and you can actually go to most stores, not even the health food stores anymore. You can find them in some of your bigger grocery stores. You can go in and buy probiotics, live friendly bacteria. Now this brings us to opportunistic microorganisms. It's location, location, location. Your normal microbiota that is being good where it should be, getting where it shouldn't be, and causing problems. Prime example, Staphylococcus epidermidis. You're familiar with our friend SE from lab. Normal skin microbiota. Usually considered commensal. I consider it mutualistic because it's taking up space so the pathogens can't attach. Okay? But what happens during a surgery when it gets from your skin into your blood? Okay? Now sometimes it stops there. Your immune system, because we all get cuts and scrapes, we've got antibodies against Staph Epi, can take care of it. But we've found that if they can get through the blood to, say, an artificial knee that's being implanted, or directly from the skin to the artificial knee, we have a real hard time getting rid of those guys. They form a community, a biofilm, and your antibodies can't get through the biofilm to kill the Staph Epi, and antibiotics have a hard time getting through there. Okay? Oftentimes, the artificial knee or joint has to be removed and put back in. So if you go into Surg Tech, make sure that you maintain your aseptic technique. Now, what if it gets from the blood into a different organism? Okay, the liver. From there, it can go to all sorts of other places, or your immune system may take hold. Okay, so that's why we call these organisms opportunistic pathogens. They're not causing a problem if they're where they're supposed to be, but if they get where they're not, they will take the opportunity to cause disease. Now, there are some organisms that if they get out of their location and get in where they shouldn't be, i.e. the blood, they don't cause a problem. It's a completely inhospitable environment for them, so those would not be considered opportunistic pathogens. Now let's talk about cooperation among microorganisms. With a very few exceptions, there's no place where you have just one kind of bacterium, one species. Okay, and in fact, we've found with an awful lot of bacteria, they have receptors for other bacteria, not you. Okay, and we've done an awful lot of these studies with tooth biofilm. That's why I say they have receptors for other bacteria, not for the teeth. Okay, so there are microbes that cannot live without the other members of their community. Okay. These are microorganisms that we haven't been able to grow in a lab. They're too fastidious, but we're moving into research into biofilms, looking at these species we can't culture independently, looking what metabolic needs are, and we're also looking at ways to disrupt these biofilms. Say, for example, on the previous slide, with this biofilm of Staph Epi that forms on an artificial joint. If we can figure out how to disrupt that biofilm, we can break up that biofilm and your immune system can have at it and get rid of them. Okay, that's a lovely thing. If we can figure out how to break up environmental biofilms. It's all fine and good to have a biofilm growing on a rock in a stream, but it's not so good when it's a water pipe. Okay. Environmental microbes will form pond scum on the inside of a pipe and clog it. Now let's talk about etiology, which is the study of the cause of disease. Now, when we're talking about infectious disease, we have to talk about Koch. Koch was living at the same time as Pasteur and was actually a competitor with Pasteur. And he came up with what we call Koch's postulates, and this is the gold standard for figuring out if a microbe causes disease. So the first thing you do is you get an organism that has the disease. Here we've got our poor little mouse that's dead of a disease, and you take blood, tissue samples, etc., and you isolate a single organism that you can observe underneath the microscope and grow on solid agar. Then you take that pure culture, just one organism, and you inject it into a healthy lab animal. If that healthy lab animal develops the same symptoms of disease that was seen in the wild population, and if you can re-isolate exactly the same organism, then you can say this organism causes this disease. Well, that works all fine and good for strong pathogens. It doesn't work so well with opportunistic pathogens because you can see it in health as well as in disease. But we can still use Koch's postulates with opportunistic pathogens by moving them from an area where they're not causing disease, and they always cause disease when you move them to an area that does have disease caused. Now, there are some exceptions to Koch's postulates where disease is being caused by multiple organisms or by an imbalance in the normal microbiota population. Okay, periodontal disease is a prime example. From all the samples that we've been able to isolate from caries, cavities, and from periodontal disease, we find that they're the same organisms that are found in healthy mouths. So it's taken us quite a while to try to figure out what organisms are causing disease. And we've found that disease is often caused by an imbalance, where you have one microbe species that is in small numbers, that because of diet, or in this case this person not brushing their teeth, they become a higher population and start to cause problems. Okay, so in situations like this we can't use Koch's postulates. So technically we can't prove, unquote, that this is causing the disease. Okay, now there's all sorts of other exceptions to Koch's postulates, but I like that one because it's nice and obvious and it's something I've worked with. Next, we're going to talk about classifying the various types of diseases. We're going to talk about occurrence of diseases, and we're going to talk about patterns of disease. Now, in the textbook, make sure you learn the terms that relate to the severity or duration of a disease and the extent of host involvement. I'm not going to go over that in this presentation because it's mainly vocabulary and you just have to memorize it. Now, when we talk about occurrence of a disease in a population, we use two different terms, incidence and prevalence. Incidence is where you take the new cases that are seen and you divide that by the total population of the area you're talking about. And then you times it by 100 to get a percent. Now, what am I talking about? For example, we track the incidence of influenza in various states. We also go down to the county and city level when we're tracking an epidemic. Okay, that would be the incidence. Now, because influenza is never chronic, we generally don't talk about the prevalence. The prevalence is all the cases divided by the population of an area times 100. Now, when you have chronic diseases, you can have incidence as well as prevalence. Let's take HIV. Once you have somebody who gets an HIV infection, and hopefully with medication, do not move to classic AIDS, you have prevalence. Okay, Prevalence versus new cases, generally we talk about it per year. So the incidence would be the cases, new cases of patients that are discovered to be HIV positive versus all of the cases, all of the patients that are currently alive. Now you can track this by state. With HIV, we have a tendency to track it by state as well as the nation, and we also track it worldwide. Okay? So make sure you understand the difference between incidence and prevalence, and make sure you can do the math. It's not too bad, but if you want to, we can certainly go over it in lecture. Now let's talk about patterns of diseases. Not everybody who becomes infected develops diseases. We have predisposing factors for disease. Couple of examples from the book. Women are more likely to get urinary tract infections. It's because we have a shorter urethra. It's less distance for the microbes to crawl up to the bladder. That is a predisposing factor. Men have a tendency, more of a tendency, to develop meningitis and pneumonia. With that one, I have no idea why. Do men breathe more? I don't know. Anyway, we find that meningitis and pneumonia shows up more often in men. So in this case, sex would be a predisposing factor. Okay? There are other predisposing factors. It can be anything from genetics to exposure to the microbes, living conditions, socioeconomic situations, occupation, you name it. Now let's talk about development of disease. Okay? You've all experienced this. All of us have been sick at one time or another. Okay, so we divide up the development of disease into a series of stages. Incubation. You have no signs or symptoms. You've been exposed. You're infected, and the microbes are replicating within you. Now, sometimes your immune system kicks in, and you never get sick. That's the optimal. Doesn't always happen that way. Now, the prodromal period is not seen in all diseases. This is where you start to feel just not well before you start getting the classic signs of the disease. Okay. Some illnesses you skip the prodromal and you move directly to the period of illness. This is where you have the most severe signs and symptoms, and this is generally where we can tell what you're sick with. Now, your immune system kicks in. We start to plateau on the numbers of microbes. Signs and symptoms start to decline. You start to feel better. You think you're not going to die. And then you get into the period of convalescence, where you have repair of any damage to tissue. You're feeling better, and the number of microbes trail off into nothing. Now, this looks an awful lot like what else? Bacterial growth curve. Incubation, we can say that's equivalent to lag time, except for lag time ends in disease before we start getting symptoms. And then this would be the log phase or exponential growth, stationary phase, decline. Okay? Make sure you're able to relate these two, because I love to ask you those kinds of questions on a test. Well, that's it for part 1. In part 2, we've got some good gross videos that I pulled off the internet, so make sure that you're ready for them. Make sure you're not eating